All right, you guys. So now we are going to talk about biological resistance, and we're going to talk a little bit about genetic drift and how genetic drift can often be measured. So we're looking at SB6 still and focusing on D and E. So we're looking at the theory of evolution. With D, we are looking at undirected changes and how those undirected changes have led to changes in populations. And then we're also going to look at how natural selection plays a role in causing biological resistance. So, our goal today is to explain how genetic drift, gene flow, non random mating, and mutations all affect natural selection and evolution, and also to explain how biological resistance is affected by natural selection. So let's start off. I want you to think about this. We're going to talk about this in class when we get back into live class. But if you've ever had an ear infection or strep throat, how was it treated? I want you to think real in depth about how was that particular infection and treated. Typically, it was probably treated with antibiotics. And there's a reason for that. That's because antibiotics can kill off the bacteria. It allows for people to be treated and people to get better because the antibiotics are able to directly attack the bacteria that is infecting your body. There's a catch to this, though. Bacteria reproduce through a process called binary fission. Basically, everything inside the bacteria doubles up and then it splits in half. This happens super, super, super fast. One bacterial cell can become 30,000 bacterial cells in under 5 hours. This is why sometimes you can be exposed to an infection and the very next day you're sick. It's because after you're exposed and after that infection gets into your body, it rapidly multiplies. And as it's rapidly multiplying, the bacterial cells can crowd out healthy cells and they can cause diseases. In your body, there's lots of things that happen in response to that infection. The one of the very first things is that your body signals the cells send out a signal that, hey, there's a problem over here. And white blood cells and antibodies just really flood into that area. This is what causes things like pain, sore throat, runny nose, fever, all of that. If you do, in fact, have a bacterial infection, doctors will prescribe antibiotics to treat that infection and kill the bacteria. So I want you to think about if the antibiotic works on most of the bacteria but doesn't kill them all. Let's say a doctor gives you a 10-day course of antibiotics, but you're feeling better by day 7, so you stop taking the antibiotics. You've killed off almost all of the bacteria, but not all of them. And unfortunately, what that does is creates potentially super bacteria cells because the bacteria then are able to evolve and create new strains that are resistant to that antibiotic. So let's see what our TED Ed video has to tell us.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere: on the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis. all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that help them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So how much time do we have before these superbugs take over? Well, in some bacteria, it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta-lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta-lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria like Salmonella even sometimes produce enzymes like beta-lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strains that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolones by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria. And although development of new antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments. Other scientists are investigating alternate solutions, such as phage therapy or using vaccines to prevent infections. Most importantly, curbing the excessive and unnecessary use of antibiotics, such as for minor infections that can resolve on their own, as well as changing medical practice to prevent hospital infections, can have a major impact by keeping more non-resistant bacteria alive as competition for resistant strains. In the war against superbacteria, de-escalation may sometimes work better than an evolutionary arms race.
All right, you guys. So that is a fabulous explanation of why antibiotic resistance happens. So, now we're going to talk about what happens if there's not any genetic change around. A population with no genetic change experiences something called equilibrium. And what that means is that nothing changes. It basically is all balanced out. So equilibrium just means balance. And these populations that are in genetic equilibrium, they're not evolving. That means they're not changing. And that is because there's no mutations, there's very large populations, there's isolated populations, nothing's moving in or out. All of the members survive and reproduce, so there's no selection acting on it, and mating is completely random in these situations. But does this ever actually happen? Not really. Because we need lots of genetic variation to happen. We want genetic variation because we want evolution to be occurring. If all the populations were the same, processes that cause evolution would have no effect because the genetic makeup would never change. So if we look here, the genetic variation is really good because We've got our first generation of beetle population, and then we've got many, many, many generations later, the same beetle population. What happens is over time, the organisms are able to have a change in gene frequency in the gene pool because the environment changed. So maybe this first generation of beetle population lives in a place where there is a lot of grasses, lots of green grasses. But then over time, those lots of green grasses begin to die off, and you're left behind with much more dirt and things like that. Well, these two brown beetles, they were able to survive because they blended in, and they're the ones that reproduced. So it changes the gene frequency. So let's check out our TED Ed video.
The Five Fingers of Evolution. A thorough understanding of biology requires a thorough understanding of the process of evolution. Most people are familiar with the process of natural selection. However, this is just one of 5 processes that can result in evolution. Before we discuss all 5 of these processes, we should define evolution. Evolution is simply change in the gene pool over time. But what is a gene pool? And for that matter, what is a gene? Before spending any more time on genetics, let us begin with a story. Imagine that a boat capsizes and 10 survivors swim to shore on a deserted island. They are never rescued, and they form a new population that exists for thousands of years. Strangely enough, 5 of the survivors have red hair. Red hair is created when a person inherits 2 copies of the red gene from their parents. If you only have 1 copy of the gene, you won't have red hair. To make this easier, we will assume that the 5 non-redheads are not carriers of the gene. The initial frequency of the red hair gene is therefore 50%, or 10 of 20 total genes. These genes are the gene pool. The 20 different genes are like cards in a deck that keep getting reshuffled with each new generation. Sex is simply a reshuffling of the genetic deck. The cards are reshuffled and passed to the next generation. The deck remains the same, 50% red. The genes are reshuffled and passed to the next generation. The gene pool remains the same, 50% red. Even though the population may grow in size over time, the frequency should stay at about 50%. If this frequency ever varies, then evolution has occurred. Evolution is simply change in the gene pool over time. Think about it in terms of the cards. If the frequency of the cards in the deck ever changes, evolution has occurred. There are 5 processes that can cause the frequency to change. To remember these processes, we will use the fingers on your hand, starting from the little finger and moving to the thumb. The little finger should remind you that the population can shrink. If the population shrinks, then chance can take over. For example, if only 4 individuals survive an epidemic, then their genes will represent the new gene pool. The next finger is the ring finger. This finger should remind you of mating because a ring represents a couple. If individuals choose a mate based on their appearance or location, the frequency may change. If red-headed individuals only mate with red-headed individuals, they could eventually form a new population. If no one ever mates with red-headed individuals, these genes could decrease. The next finger is the middle finger. The M in the middle finger should remind you of the M in the word mutation. If a new gene is added through mutation, it can affect the frequency. Imagine a gene mutation creates a new color of hair. This would obviously change the frequency in the gene pool. The pointer finger should remind you of movement. If new individuals flow into an area or immigrate, the frequency will change. If individuals flow out of an area or emigrate, then the frequency will change. In science, we refer to this movement as gene flow. All 4 of the processes represented by our fingers can cause evolution. Small population size, non-random mating, mutations, and gene flow. However, none of them lead to adaptation. Natural selection is the only process that creates organisms better adapted to their local environment. I use the thumb to remember this process. Nature votes thumbs up for adaptations that will do well in their environment and thumbs down to adaptations that will do poorly. The genes for individuals that are not adapted for their environment will gradually be replaced by those that are better adapted. Red hair is an example of one of these adaptations. Red hair is an advantage in the northern climates because the fair-skinned allowed ancestors to absorb more light and synthesize more vitamin D. Thumbs up. However, this was a disadvantage in the more southern climates where increased UV radiation led to cancer and decreased fertility. Thumbs down. Even the thumb itself is an adaptation formed through the process of natural selection. The evolution that we have described is referred to as microevolution because it refers to a small change. However, this form of evolution may eventually lead to macroevolution or speciation. Every organism on the planet shares ancestry with a single common ancestor. All living organisms on the planet are connected back in time through the process of evolution. Take a look at your own hand. It's an engineering masterpiece that was created by the 5 processes I just described over millions and millions of years. Can you recall the 5 main causes of evolution from memory? If you can't, hit rewind and watch that part again. But if you can, give yourself or your neighbor a big 5.
All right. So just a quick recap of what those 5 fingers of change are. The pinky finger is shrinking populations representing genetic drift. The ring finger is non-random mating because most of the time organisms choose their mates. The middle finger represents mutations. The pointer finger represents movement position like gene flow. And then the thumb represents natural selection and adaptation. So I want you to be thinking about what kinds of things can we get from graphs. So we can look at a graph, we can look at the names, and the name tells us information about what the graph is about. So what's America snacking on here in this first graph, or the most popular music genres in the second graph. And you can see they even include years in 2017 or in 2013. And then these bar graphs tell us what were most Americans, at least the ones polled, snacking on, or what did they think was the most popular genre during those time frames. So you can use graphs to get lots of important information for yourself. And then we need to talk a little bit about Hardy-Weinberg. So Hardy-Weinberg is a principle that states that a population's allele frequencies will stay constant unless one or more factors like mutations or natural selection cause those frequencies to change. So if a population is in equilibrium, then it's also considered a Hardy-Weinberg population because nothing's changing. That's very rare that that happens because most of the time populations are not in that equilibrium. We can use this Hardy-Weinberg principle to predict allele frequencies in a population's gene pool. As far as the math goes, you do not need to know how to do it. You do, however, need to know it exists. And then you've also got to be able to analyze graphs and charts. So let's take a quick view of Hardy-Weinberg.
Do you like math? Sometimes when people are starting out in biology, for some reason they assume there is no math in biology, but then they soon find out they were wrong. My math teachers were telling the truth when they said math isn't everything. Chi-squares, osmotic pressure calculations, standard curves with gel electrophoresis, Punnett square ratios, these all have math. But one of my favorite topics that involves biology and math and is also absolutely fascinating is the Hardy-Weinberg equilibrium. It got its name from a mathematician and a physician, and it states that a population's allele and genotype frequencies are constant unless there is some type of evolutionary force acting upon them. A reminder from our ecology videos, a population in this case is a group of organisms that are all the same species and can breed with each other and have fertile offspring. However, there can be variety among a population as they are not clones of each other. But based on Hardy-Weinberg equilibrium, the population's allele and genotype frequencies would remain constant if no evolutionary force acts upon them. If we have these frogs here, assume they are all the same species in this example, but there are some slight differences. In our simplified example, some of the frogs are lighter green and some are darker green. We're going to use a simple genotype in this example. All frogs in this population will be either big G, big G, big G, little g, or little g, little g. there's also an allele frequency in our population. We're going to say here that 60%, a frequency of 0.6, of the alleles are big G. 40%, or a frequency of 0.4, of the alleles are little g. Notice the percentages add up to 100%, and the frequencies add up to 1. Now, to be in Hardy-Weinberg equilibrium, we have to have 5 assumptions. Number 1, no selection. No natural selection is acting upon these frogs. That means neither dark green nor light green will have any impact on reproductive fitness. Number 2, no mutation. Baby frogs inherit genes from their parents and there are never mutations. Number 3, no migration. Frogs can't come in, frogs can't go out. Number 4, large population. There are a lot of frogs. It turns out small populations are more vulnerable to genetic drift, by the way. See our genetic drift video. Number 5, random mating. The frogs mate without any specific choice. All of these 5 assumptions must be kept in order for Hardy-Weinberg equilibrium to happen. So, in real life, does this generally happen? No. For example, in real life, maybe in a certain environment, predators can more easily see the light green frogs, so perhaps they are eaten more and have less reproductive fitness. So,
All right. So as you can see with Hardy-Weinberg, it's all about things not changing. And so you can usually tell if a population is in Hardy-Weinberg equilibrium by going through and doing a bunch of the math stuff, and that will let you know. Um, we're not going to get into all of the math portion of that because that is not something that you need to be able to do for this biology class. So that's everything that I have for you, though. I hope that you have a wonderful, wonderful day, and I will see you in our next class session soon. Bye, guys.